Good evening to one and all. At the outset, I warmly welcome all the participants for the session 2 of 2-week blended FTP on radiation exposure and its impact on human health. So the session expert of the session 2 is Professor Dr. Surjit Debanath. He is going to give a lecture on the clinical applications of radiation assessment through computational simulation. He is the head of the Department of Medical Laboratory, Technological Women's Polytechnic, Government of Tripura. Now I hand over the session to Sir.
Okay, thank you. So first of all, I would like to thank AICTE ATAL for providing an excellent innovative platform where interactions of thoughts and research process as well as academics and continuous learning, teaching, all these kinds of discussion can be done in an excellent way. My sincere gratitude to this thing also, the coordinators also of this FDP. So today I'll be speaking about some clinical application of radiations. Now, before going to the discussion, let us understand 2-3 different things. I am absolutely sure that since all of you belong to the classical science background, that is physics and chemistry, you also have opportunity in last couple of days to understand the very basics of radiation exposure and all such things. Now, in my today's discussion, I'll divide my whole session into 2 parts. In one part, we will have a discussion on what is the beneficial aspects of radiations. Now, these aspects, all of us already are aware about it. For example, if you are having an what do you call water purifier, you use radiation there in order to to kill the germs that is present in the water. A long back, there was also a process which is now a standard norm in food tech companies. Food tech companies means there are companies which will provide cooked food or fresh food to you, to the customers. They are also using radiation exposure for sterilization purpose. So this is one part. One more part are non-ioning radiations, where we are talking about positron emission, then computed tomography and all such things, MRI also. So there are 2-3 things. Now, one more aspect is the aspect of modulation in genetic behavior. So this is my interest. So I belong to the fraternity of biomedical sciences and technology, where my profound interest is on the impact of radiation on DNA. So whenever we talk about this, there are 2 aspects of it. One can understand that we are only talking about how cancer produces. This is fine, but eradication of cancer is also done through radiation. So both the aspects we will study or we will just discuss. Okay. So coordinator sir, I am audible, no? And English is understandable?
Yes, sir. Yes, sir. Yes, sir.
Okay. So what I'll do now, I will share one link and I'll request our coordinator sir to please click on the link, and it will take us to a video. And for the time being, we'll just see the video, and sound will also be there. But why I'm playing this video, I will tell later on. Okay. So I'll stop sharing now for some time, and I'll go to the chat box and I'll share the link. So anyone of you can this thing play, but I'll request our coordinator sir to please click this link and play this video and share his screen also, just to break the ice, and it will help us to have some more serious discussion later on. On a lighter mode, I'll request our coordinator sir to please play this particular video for all of us. Subsequently, we'll move on to the next main part of the discussion.
Professor, now shall I share the screen, sir?
Please.
Thank you.
Okay, full screen, sir. Full sound.
Yes.
Okay, coordinator, sir. This much will be enough. So we'll just, okay, close, sir, close. Okay, now I'll go to my this thing discussion. We just had a video, and now we'll start our discussion. We can switch off the video now. Okay. So this was our slide. Okay. So there is the United Nations Scientific Committee on Effects of Atomic Radiation. So it's a prime body which looks into many things. So it looks into the areas of works of this organization is which kind of weapons are being tested, and then what are the medical exposures, and then occupational exposures, and then public exposures. They also look into generation of electricity through radioactive materials, and then natural radiation sources, which is very important aspect of study for many researchers throughout the country, even in my area, because it has been identified that in buildings, in sea beaches, even in this thing, food grains also, there is huge contamination or huge exposure of radiation, which ultimately leads to multiple bad effects on the health. Then subsequently, there are many more things. Then this particular organization will also do impact assessment on children. Then what are the biological effects and mechanisms? What are the health effects? It also looks into the possibilities of nuclear accidents and many such things. So why this body is there? Because it is established fact that radiations are having negative impact as well as some positive impact. Now, what is the role of teachers? So first of all, we are teachers recruited by AICTE at various capacities, and our primary job is to teach our students. Now, while we will be teaching our students, we should also impart both sides of the knowledge. What is the pros and what are the cons? Now, while doing so, we should more look into the beneficial aspects of radiation and let them understand that there comes with huge power comes a lot of responsibility. And therefore, this particular organization, United Nations Scientific Committee on Effects of Atomic Radiation, advocates that it should be included in the curriculum and students should be taught in an ethical way how to use radiation for betterment of the human race. So these are some equipments are very much aware from your side, which will help us to understand what is the what is the magnitude of exposure of radiation at various points. It could be inside a building, it could be inside a building which is made up of what we call cements and particles which is contaminated with radiation. It will also help us to understand if there is radiation exposure in the river side or in the sea beaches. So it's a handheld device which can help us tremendously to understand what is the exposure we are going through it. Now, what are the beneficial aspects of radiation? Medical diagnosis and treatment. This is a very important aspect where we are working in a significant way. In our lab, we are trying to classify PET scans and CT scan to make a machine learning algorithm which will help us to reduce the burden of human intelligence. Now, just to give an introduction to all of you, human intelligence is very, very costly. You can also understand if you do an X-ray, you may not understand the intricacies of the whole X-ray plate. So you will go to a doctor and he will he will give you some information that what has gone wrong into your internal tissue. If it is a hard tissue, then a common X-ray will do. But if it is a soft tissue, then problem arise. And if there are some abnormality in the heart soft tissue, then the problem becomes manifold. In those cases, we need to work with different high level devices, which are positron emission tomography, then CT scan, and for magnetic imaging resonance, and so on and so forth. So this is a very important part of radiation, where radiation exposure is there, but it helps us for mitigating the immediate what you call emergency that has happened. For example, one trauma patient has come, the internal bleeding is there in the head or inside the brain, there are some hemorrhage or there are some blood clots. How you will detect? Nobody can detect other than a CT scan. No one can. There is no other device. There is nothing which can help you in this case. So you have to go for CT scan. Many a time CT scan comes with two things: either there will be a contrast or without a contrast. So there are two aspects of it. Both of these things are bad. There are some bad effects. So if there are some bad effects, we will not do. No, to save the life, we have to do CAT scan. Then research and application. Research and application, tremendous. This thing is going on everywhere, in classical science as well as in application-oriented science that are developing day by day. Industrial and manufacturing applications, as I have given the example, all of you are very much aware that ultraviolet ray, what it does, it will kill the all kinds of germ, even the viruses, because what happens now, viruses are made up of nucleic acids. All viruses, for example, corona, very famous now, everyone knows. So what is corona? Corona is nothing but a virus which is made up of RNA. RNA is nothing but a polymer of a nucleotide which undergoes degradation when there is an impact of ultraviolet ray. So ultraviolet ray is it is having, because the energy score is very, very high, since the wavelength is very, very low. So what happens in inverse, it is the impact of the energy is very high. So what it will do is it will degrade the DNA, even RNA, and eventually it will cause the breakdown of the nucleic acid, and eventually the bacteria, viruses, and all living organisms will die. Okay. So industrial application and manufacturing, this particular application is huge. It is not very tech savvy like CT scan, MRI, and other kinds of scans. It is very easy to deploy, and multiple different devices can be what you call originated. You can even patent some devices by deploying ultraviolet ray based handheld devices. It's very easy to do, not much innovation is needed, but your patent is guaranteed, because so much of application is there, isn't it? So then what else? Consumer products, safety and security, spacecraft power supply. These are some. I have very less information about all these things. Electric power generation, that is also there. So the first video that we have already we have demonstrated, it has something related to this particular last point, electric power generation. We'll come to that. Coordinator sir, just let me know whether my voice is clear and you can see and you can also understand. So the English is clear for everyone?
Sir, it is very clear. Thank you.
Okay, absolutely. Okay, thank you. No, my pronunciation may be a bit different from yours. So what exactly happens is characteristics of nuclear imaging modalities. So these things are also there, and you already have sufficient information about all this. So here there is an important aspect I would like to share. There is a protocol in biomedical sciences which is called targeted therapy. So let me tell you what is targeted therapy is. The most important aspect of beneficial aspects of radiation. So all these days, what has happened in radiation therapy? No, the radiation cannot detect the exact location where neoplasm is there. Neoplasm means the tumor. Now there are two kinds of tumor. One is benign and another is malignant. In benign tumor, what happens? The tumor is localized. There is a particular area where the tumor is there. But in malignant cases, what happens is the tumor doesn't have any boundary. It can go anywhere, it can do metastasis, and it can even go to some other unrelated tissue. Give you an example: breast cancer, the very important group of cancer that we study in females. So now this is very interesting. What happens is there many cancers are there which are region specific. Okay, region specific. So this kind of people are having exposure to chewing tobacco. So they will have what? Oral cancer. This kind of people are having living in a area where high contaminated air is there. So they will have lung cancer. Okay. Breast cancer is like omnipresent. It can be anywhere. There is no reason. Nobody can predict that where it can occur. So in this case, what happens? Breast tissue is a limited area tissue, but the problem with this is rapid metastasis. Metastasis matlab kya? It will originate in a single cell and then it will spread throughout the tissue. It will destroy the tissue itself and then move on to some other tissue, lung, pancreas, and then what else? Bone. Even it will go into bone metastasis also. So it's a dangerous kind of cancer. Now, in those cancer, the problem is you cannot, the radiation exposure is generalized. If you cannot, because you cannot, what you cannot do is, the precision is very less. All these days, precisions are very less. Whether it is guided, non-guided, most of the time, radiations are non-specific. So it will destroy the DNA of the cancer tissue. At the same time, it will also destroy some good tissue. So there is a rapid, what you call, contraindication. Contraindication, if somebody is there who is having a internal pacemaker, so he or she may not take this particular radiation. Okay, so multiple different contraindications are there. So what is the way out? The way out is targeted therapy. Now in targeted therapy, what happens? In targeted therapy, what happens? They will be giving you a systemic dose. Systemic dose means एक injection देगा, लेकिन injection का जो injection में there is a nano carrier which will carry the radioisotope, and that radioisotope will not expose anywhere. It will expose because it will be coated by a layer of polymers which will only get exposed at a particular location only. And so what will happen? The radiation exposure will be there on a particular kind of cell, not everywhere. So this becomes a very interesting therapy, and throughout the world, biomedical scientists are working on this. So I'll also demonstrate a small video which has been produced by Monash University, Australia. So you will see how specific the design is. These are these are in lab scale, not in a clinical trial scale. But we are anticipating that in next 10 years, it will be a regular thing. Okay, so targeted radiotherapy will be a what you call reality in next 10 years. So we'll come to those points. These things you already know. I will not discuss. So these are some also other what you call radioisotopes with medical applications. And as you can see, what is the ultimate production and what is the emission type. The half-life is also there, and the Emax is also been calculated. So here, as you can see, the highest half-life, lowest half-life is by iodine 131. Is 131? Yeah, iodine 131, and this is the highest half-life, most stable one, and the least stable one is. As you can see here, the least stable one is fluorine. Okay, so these are different what we call characteristics of potential radionucleotides for tumor imaging. So this is useful for the imaging purpose only. Okay, so there are some other examples of the malignancies where MRI is routine. Okay, so in this case, what happens? MRI is effective on soft tissue, and it can detect some specific tissues very nicely. On a development of cancer, very nicely it can what do you call detect. For example, brain. So in brain, what happens if we use MRI along with this thing, what do you call radiation exposure, the better delineation of the brain tumor on MRI is possible, as these what do you call uh, because what happens in a CT exam, there are some difficulties, but in this case what happens if um, MRI is used, so what happens, the boundary of the cancer can be nicely detected. So many such examples are there for prostate cancer, for nasopharynx. Nasopharynx means oral cancer. Basically, oral cancer takes place where? In the pharynx, the deeper, not exactly on the outer face, but mainly it originates from the deeper end of the nasopharynx. Then there are liver and pancreas, that is GI tract. GI tract cancers are also very common in India. Then, but here there is a point. This GI tract cancers are basically originated due to impact of viruses. If virus exposure is there, for example, hepatitis B and C, then cancer promiscuity is very high. But in normal cases, liver cancer may not be very frequent. Then there are spine cancer as well as prostate cancer. In males, prostate cancer is very common. Now here what happens, we'll see the impact of ionizing radiation. Now this ionizing radiation, what it will do is, it may directly impact the DNA, and it will lead to a single stranded break. As we all know, DNA is the molecule which will govern the ultimate what do you call architecture of the cell. So what exactly happens is, ionizing radiations can directly affect the DNA, and it will cause a single stranded break, or through water molecule, by forming oxygen free radicals, it can also cause rapid damage, and it may impact both the strands. Now this particular thing is very interesting, because here what happens, no, ionizing radiations are useful for eradicating the cancer, because in cancer cells also DNA is there. In cancer cells also DNA is there, and that DNA needed to be degraded. Now this is the idea of radiotherapy in cancer. In radiotherapy, what exactly happens? A particular kind of radiation is given, so that the cancer tissue is exposed to that radiation, and ultimately it starts decreasing in size. Why decreasing in size? Because all the cells starts degrading due to the impact of the radiation. Now, this particular ionizing radiation can cause damage in the single strand as well as in the both of the strand. Both are damaging to the what you call DNA. Then there are some other characteristics of potential radionucleotides for tumor radiotherapy. So multiple different radionucleotides are there, which will help us to eradicate cancer. But as I have said, till now it has been very what do you call non-specific, and therefore only in some cases cancer eradication. In some cases, radiotherapy was excellent, but for breast cancer, no, the cases are very mixed. Not very good results can be found, because the location of the cancer is such that multiple different organs are associated. For example, if there is a head and neck cancer, there is a kind of head and neck cancer. In that case, radiation exposure will work well, because it is not exposed to the rest of the tissue. But in breast cancer, what would happen? Many organs are there in that area, so there is a high possibility that other organs will get highly damaged, and therefore multiple different problems will arise. The patient will lose hair, the patient may lose teeth, sometime eyesight may go, skin will become rotten, and multiple different abnormalities will happen. So nowadays what the scientists are doing is, they are targeting the tissue through a nano carrier. So what they are trying to do is, they are trying to make a nano emulsion and then injecting it, and the emulsion runs towards the bloodstream and enters into a particular tissue. We'll have a demonstration of this. Now there are some what do you call very good examples where some examples has been given excellent results with respect to a particular radionucleotide. As you can see here, lutetium is very important for neuroendocrine tumors. Now, what are neuroendocrine tumors? You will be amazed to know there are tumors of pituitary gland. Now, it's a very small part which is present inside the what do you call brain. Now, if there is a cancer in pituitary, the person will not instantly die, but it will die a very painful death. Similarly, there is another endocrine organ which is called adrenal, which is present near the kidney. Now, adrenal cancers also are very, very dangerous. Okay. Now, in these two type of cases, a particular kind of radionucleotide is very, very effective. Then there is iodine, which is very helpful in thyroid cancer. Now, thyroid cancer is also very interesting. In thyroid cancer, what happens? Thyroid is the organ which regulates the iodine balance in the body. So, thyroid gland, it makes a hormone which is made up of iodine itself. Okay. So, what happens? The cause of thyroid cancer is also iodine radioisotope, and the what do you call medicine for thyroid cancer is also iodine radioisotope. So it's a very this interesting feature of this thyroid cancers. Okay, so you can, if you want, you can take a screenshot of this. The which radionucleotide is important for which kind of cancer. Then subsequently there are more this thing what do you call examples where the characteristics of beta emitting radionucleotides that have been investigated for in situ delivery of radiation therapy. Now, in situ in situ delivery of radiation therapy is where a, a, a catheter is inserted into the body and radiation is given to a particular area. So we'll have a little discussion. I think many of you are already aware. So there is a new kind of guided therapy. In this guided therapy, what happens is one catheter is inserted. Now that catheter you are aware of. For example, this same catheter is used for angioplasty and angiogram. So if somebody goes for an angiogram, what exactly happens? From the leg, no, one vein is cut. Now through this vein, and the blood is stopped, and blood circulation is stopped in that particular area, and then one catheter is inserted. On the top of the catheter, there is a what do you call optical fiber, which will see what exactly happens, and there is a small miniature camera onto it. So the doctor will able to see what exactly happening near the heart. So through leg it will enter and it will come up to the heart. So it's a long way to go. So through this kind of channeling devices also, these days radiotherapy are provided. Now this is the example of radionucleotides which are suitable for catheter guided radiotherapy. You will see a little demo of it, how exactly it looks like and what are the cases where best possible results can be given for catheter based radiotherapy. These are the catheter based radiotherapy. Oh no, these are not the catheter based radiotherapy. These are the thing that we are working on. What exactly happens is there is a kind of for example, there is a tumor somewhere inside the deep inside the body. So it is a malignant tumor and it cannot be operated. Uska operation nahi ho paye. So in that case, what would happen? The radionucleotide should be given. Now, what exactly happens is at room temperature, these radionucleotides are separated. They are not coming together, and therefore the exposure is not the impactful. Okay. So what happens is there are some polymers, basically protein molecules, which are connected to these radionucleotides. Okay. So when they are at room temperature, nothing will happen. Okay, basically these will be preserved in low temperature, for example 4 to 10 degrees, and in room temperature there will be a thaw, and after a little bit of acclimatization, then we'll inject it to the patient. Now in patient's body, approximately temperature will be 30 to 37 degrees centigrade. Okay, now at room temperature there will be no polymerization. Okay, but when it reaches body temperature, that means more than 30, near 30 to 37, this range. So what would happen? These proteins will come together. Now when these proteins will come together, the radiation becomes impactful and it starts producing its effect. So it is one kind of a guided one, not the best possible way. Although this is not the best possible way, we will demonstrate one video where you will see the best possible mechanism of guided radiotherapy. So this is also good because what happens is it is also going inside the body and then doing the exposure. So it is helpful for the therapist. Therapist matlab kya? It is helpful for the clinician, for the doctor, because when it is present in the room, no, it will have no problem, no exposure, no this thing will be there. Okay, the mechanism is such that when aggregation will take place, the radioisotope will become impactful and it will start providing its what you call biomedical effect. Acha. Now here there are some other mechanisms also. Please see. So what exactly happens is directly on the tumor, if we inject, then what are the different what you call mechanism that happens? So there is a what you call radioisotope based nanoparticle. Okay. Now these nanoparticles can be made up of anything. It could be made up of gold. It could be made up of silicon oxide. It could also even be made up of iron. Okay. Now when we'll inject it, and then radio labeling will be done. Now then once they are radio labeled with this kind of with this kind of radionucleotide, then they becomes radio labeled nanoparticles. Okay. Then they will be injected in the body. Now once inside, what will happen is they will come closer to the cells. Now these cells are tumor cells because we have injected the molecule directly onto the tumor. Because what happens, this tumor is at least localized. Okay, metastasis has not yet started. It's an early stage tumor. Now here what would happen, these molecules will go and destroy the cells. Now cell destruction takes place in which mechanism? We have already understood. Ionizing radiations, what they'll do, they'll directly impact in a single strand, or if it is through the oxygen free radicals, then it will break both the strands of DNA. Now, these are some other isotopes which are commercially available. Gamma emitting what do you call radiation, and some very good results are there, and these are commercially available also. So the half-life is approximately 64.2 years, and subsequently the material is a resin. So what happens is it's a resin-based material. Resin is nothing but an inorganic polymer which will entrap the radioisotope, and then it will be injected into the body, and subsequently it will do its what do you call effect. Now what is the effect? It is effective against carcinogenesis of liver, where metastasis to intrahepatic areas has been taken place. That means the liver cancer has infiltrated many tissues. So here you can see, here what happens, a catheter is inserted, and from which the radiation tagged particles will come, and then it will enter into the circulation and kill the particular tumor. Now this is some amount of guided radiotherapy. Now these are very specific examples of radiotherapy, which is also called in situ radiotherapy, where what happens is, as I have said, that there will be a catheter which will take the what do you call radioisotope based particles or gels into a particular organ. Now inside the organ, it will do its all work and subsequently undergo disintegration. Okay, although it will not get completely disintegrated, but at least some amount of disintegration will take place, and eventually what would happen, the cancer becomes limited. If it is not completely eradicated, it is becoming limited. And this is one more type where prostate cancer is being tried. So previous one is a liver cancer, and next one is a prostate cancer. In these two cases, this kind of in situ radiotherapy means either a catheter means directly inside the tumor, not systemic. In systemic chemotherapy or radiotherapy, what happens, pura body ke andar chala jata, so it will enter into a particular area. If it is still there, the cancer agar bahut zyada phail gaya, then it may not be very impactful. But if it is in the earlier stages, then what would happen, it will help us to what we call control the further metastasis. So these are again some mechanisms. So how exactly cancer happens, this also is there, and how they are degrading, this is also there. So what happens is the radio labeled nanoparticles you can see in the blood vessel. The blood vessel is, you can see the brown color boundary, no? So inside the boundary, some yellow spots are there. Can you see? Now these are the radio labeled particles. Just outside to that, there is a cancer. Outside the outside the blood vessel, you can see there are two cancers. Okay, two groups of cell. On top there is a small group. In bottom there is a large group. Now what would happen? These radioisotope based nanoparticles, they will infiltrate from the blood vessel and will enter into the tissue. Now inside the tissue, it can do two-way mechanism. Please see, there is a active tumor killing and there is a passive tumor killing. In passive tumor killing, what happens? The radioisotope which is entrapped into a sphere, it will enter into the, it will deliver its content near the cell, and the cell will start to degrade. As you can see in the bottom, no, tumor cell is shown in the blue color. Then there is endothelial cell. Now what is endothelial cell? Endothelial cell is the blood vessel through which you are putting that radioisotope. Okay. So what is happened in the passive radiotherapy? The contents are delivered directly to the cell. But in active radiotherapy, as you can see, in active radiotherapy, the radioisotope which is coated by a particular material, resin or some polymer or some protein, it will get inside into the cell and then it will expose. So what happens? The chance of killing good cells is very, very limited. In passive radiotherapy, what happens? There is a chance that the radioisotope will be delivered to the nearby good cells and the good cells will also die. But in passive therapy, what happens? It is more targeted than the active therapy. In active therapy, so many good cells will also die. But in passive therapy, so many good cells will not die. At the same time, what would happen? It becomes more and more tough to reproduce. Means what? It is more costly. The drug delivery is very, very costly. So हम उसका एक video देखेंगे. Now here also you can see a little bit more of the mechanism part. How these radioisotopes are killing the cells. Okay. So there are 2-3 things. The Auger electron will produce a kind of radiation in kilo electrovolts, which will directly kill the cell. On the other hand, it will not able to penetrate into the tissue. But some particular what we call radioisotopes, they can penetrate into the tissue and even kill the cancer which is present beyond the blood vessels, which is far, far away. But in both the cases, what exactly happens is the design of the carrier is very, very challenging. So we shall see a video where a very good design has been given. Very nicely they have given the design. So I'll be sharing with you one more video which will help us to understand how targeted radiotherapy works. So I'll once again request our coordinator sir to what do you call share his screen.
Sure, sir.
Just hold on. Okay. I'll just share with you. Now, in this video, which has been prepared by an Australian university, it is only about their research, but it is very, very encouraging. So it is so encouraging that one can see how easily it can be done. But it will take a little time to execute all the steps. Till now, it is in developmental phase only. Okay. Please share your screen, sir, and play the video in mute condition. This time it
Sir,
will be mute.
Okay, sir. Sir, I got two links, sir. To which link to be open,
No,
sir?
the second one. The second one.
Okay. Sir, whether the audio should be muted, sir?
No, audience, let them speak. No problem. If some doubt they can ask.
Yes, once again, sir. Yes,
Only the video will be mute.
Okay, sir.
Let the video should be mute. So till now, what is the take home this thing? There are many different radioisotopes which has been standardized as per need. One for brain, one for one for bone, one for breast. Multiple different radioisotopes has been standardized for different types of cancer. Also, there are some good radioisotopes which are helpful for this thing also, for imaging purpose also. Now, this video will give us an idea how targeted radiotherapy can be achieved.
Shall I play the video, sir?
Please, sir. So, in this video, we will see that how targeted nanotherapy can be, what do you call, achieved. Now, this is a microscope. Please pause, sir. एक second के लिए pause कीजिए. Pause. Now, this is a fluorescent microscope. In fluorescent microscope, what happens? A cancerous cell and a non-cancerous cell can be differentiated. In fluorescent microscope, as all of you know, what exactly happens is we are having some fluorescent tags. Now, these fluorescent tags are connected to an antibody. Now, these fluorescent tags are two types. One is called rhodamine, which is red in color. Another is called fluorescein, which is blue or green in color. So now what we do is we use different types of antibody to identify which cell is cancerous, which cell is non-cancerous. Now you can ask me, sir, how can we differentiate between cancerous cell and non-cancerous? Okay, so at a molecular level, there are some signatures of cancerous cell and some good cell. In good cell, there will be only one nucleus. But in bad cell, that is cancerous cell, multiple different nucleus will be there. Now, why is this so? What is cancer? Cancer is nothing but uncontrolled divisions of cell, as all of you already aware of. So what exactly happens is, in cancerous cells, there will be multiple nucleus, means multiple DNA packets. But in normal cell, there will be a single DNA packet only. So based on multiple different factors, we can differentiate between cancerous cell and non-cancerous. Fluorescent microscope can also do the same. Now, this is a real-time fluorescent microscope where we are trying to differentiate between good epithelial cell, skin cell, and bad epithelial cell. So we will play the video now. Sir, please play now.
Sure, sir.
Now we will go into a animation. Now this is real. Now we will go to animation. It is still now real. This is also real. This is also real. See, यार थोड़ा pause कीजिए sir. 1 minute के लिए pause कीजिए. 1 minute, please pause. Now here what happens, as you know, most cells are rhodamine. Rhodamine मतलब क्या? Red cell. मतलब क्या? Pink cell. मतलब क्या? Good cell. One cell is there which is fluorescein. Fluorescein is a protein which will underglow, which will start giving fluorescent signals when projected by ultraviolet ray. So ultraviolet ray will stimulate, and then what will happen? The electrons will jump to the higher energy shells, and it will, in turn, it will produce visible light. So what exactly happens is, one is red light. Red light is good one. Blue light is bad one. Okay. Now in blue light we have to target. From now on it will be all animation. Your real nahi hoga, wo cartoon mafik hoga. Now please play, sir. Now up to this much we know. Till now we have done it. From now on, what is there going on? Abhi jo aayega next time, these are not real. We have not achieved it, but there is a high possibility that we will go there. Now please play, sir. Now we will enter into the cell. Now we'll this is the cell. Okay. Now what had happened? We have delivered one. Now this is the microsphere inside which radioisotopes are there. Whatever radioisotope you want, you can try at first with cell lines, and subsequently now you see here, you see this is designed in such a way that it will only touch the cancerous cell. देखिए cancerous cell का जो receptor है वो उसको internalize करेगा. It will enter into the cancer cell only, not in the bad cell. Now these are some receptors which will capture the microsphere and take it to the nucleus. So what happens, as you all know, nucleus is present inside the deep side of the cell. So in order to go there, there is a vehicle needed. Now this is the vehicle. Now it is going into deep part of the deep part of the cell. This is the nucleus. Can you see the blue color sphere? This is the nucleus where DNA is there. and now it is going into the DNA. Now, after going into DNA, no, it will burst. It will burst means what? All the resins will dissolve. Now, these are also biological resins. The resins will dissolve and the radioisotope will become what? Excited. Okay? Because room temperature is there. And now radioisotopes are getting delivered. And then they'll disrupt the DNA and kill the cell. Now, here you see, now only this cell will become degraded. See, it is getting what? Destroyed. Small parts it is becoming and it is only destroyed. No other cell is affected. Can you see? Now, this is the height of the story. Now, this is far. We are talking about a time like 10 years from now. Okay. So now we will once again go. Okay. Okay, sir. Now let me share now. Thank you, sir, for this coordination, sir. Okay. Now you cannot start screen sharing. Okay. You have to stop, I think, sir.
1 second, sir.
Okay. Now ready. Now ready. So what happens? Oh, नहीं ऐसा तो नहीं होगा. Close. Share screen. So what is the story? The story is that there are multiple different what we call radioisotopes that one can standardize. So if you have capability, you can standardize by working on cell lines. Now the cell lines we can provide, but we cannot provide you the radioisotopes. Radioisotopes you have to bring. Okay. So we can do the biomedical part. Okay. Now we are doing a simulation. Now this simulation, why this is important, I tell you. So which kind of damage a particular what do you call radioisotope will do, and what is the extent of that damage? How can we predict? So how many of you think that if there is a mechanism through which we can at least understand through a computer program what is the impact of that radiation? Is it good or bad? You tell me. It is definitely good because many of you are muted, so you cannot able to. So what happens? What I'm trying to say is, for example, there are studies which have given us idea that which kind of iodine 131 is good for thyroid, and lutetium or rubidium is good for some other kind of what do you call cancer. Now, how can we customize? There is a need of customization. How can we customize with age, with race, with height, with body weight, with depth of tissue? Can we able to do some kind of standardization? For doing all such standardization, if there is a simulation, it is of great value. Okay. So in our lab, we are trying to simulate and to understand how a particular kind of radiation can cause damage, and can we able to predict the damage? So these are the whole this thing that we are interested in within our small capacity. So for that, what are the mechanism? There is a very simple mechanism which is called supervised learning mechanism. In supervised learning, what happens is, I am not going to the details. In supervised learning, what happens? It is similar to human learning. In human learning, what happens? In human learning, by your life experience, you learn and then you take decisions. Similarly, the software will also take decisions based on its own experience. Which kind of experience you are giving it? So you are saying 10 times that iodine 131 is good for thyroid. So whenever it will see iodine 131, it will see that it will say, okay, you use this iodine 131 for thyroid, not for something else, isn't it? So if we are having huge amount of data, do you think that your prediction will be more precise? Your prediction will be more precise. So what is the application in radiation therapy? We will come to this point. So here we are integrating multiple machine learning algorithms to understand how can we able to predict the impact of radiation, which is your interest, which will help us to devise newer strategies. So we'll give you a little demo also for this, so that you can also able to appreciate that how good or bad the impact will be. So For
So
this, we have.
dear sir and participants,
Aha.
so it's time for a short break, sir. Let us take a very short break for 10 minutes.
Okay, okay, okay,
Before
okay, okay.
leaving, let us have a brief introduction about a resource person, Dr. Surajit Devnath.
It's a honor to have today's resource person, Dr. Debnath. He is currently the head, Department of Medical Laboratory Technology, Women's Polytechnic, Tripura, with more than 13 years of experience in biomedical research and high-level teaching. Dr. Debnath participated in various academic work in institutions of Germany, Denmark, and Hong Kong, resides in Prima Institutes, Indian labs such as AIMS, IIT Kharagpur, IIT Guwahati. Debnath is a recipient of Young Scientist Award from different agencies, a recipient of Tata Trust Grant from CME course in Royal College of Pathologists, London. Along with regular academic activities, he is also engaged with several biotechnology companies in northern and western India as a consultant. He have conducted various workshops, talks, acted as a session chair in international conferences sponsored by national and international academic agencies. Currently, he is guiding a group of international students for their internship from Russia, Turkey, and Western African countries. Dr. Debnath filed 2 patents in 2020 to 21 in India on biotechnology. It's a pleasure to have you, sir.
Okay, thank you. So we'll have a
Dear
10
sir and
minutes break.
participants, let's join back at
Okay.
8:10 p.m. Participants and the resource person, do not leave the session. Let the session be alive. We'll join back
Okay,
at 8:10 p.m. Thank you.
Okay. So can you start?
Yes, Professor, please.
Okay. Okay. So what has exactly happened? Till now, we understood that
Professor, please,
there
can
are.
you share the screen?
Yes, yes.
Thank you, Professor.
Till now, what we have understood, we understood that there are some radionucleotides which can be targeted with in situ radiotherapy as well as by means of nanoparticle based delivery system. Now, one more interesting feature of this is there is many different aspects of radiation applications that you will be studying in coming days. But in our lab, we are trying to understand that is there a mechanism through which we can predict the impact of radiation on DNA. So for that, we will give some specific demonstrations and hands-on. So all of you may not be interested, or some of you may be interested in the hands-on. Now, this hands-on is based on a concept called supervised learning. In supervised learning, what happens? We will train an algorithm to understand some problems and accordingly suggest some solutions. So now, by saying so, I'll start my what you call hands-on session. Okay. For this again, I'll request our coordinator sir for do some coordination from their end. So before going to that, I'll be okay. Let me share the links first. I'll go to the this thing, so that one more time I need to unshare. Okay. So I'll be giving 3 links all together in the chat box, and I'll request our coordinator to click on the links and help us to do the hands. Okay, coordinator sir, as you can see, we have given 3 links.
Yes,
So I'll
sir.
request you to first click on the first link
Okay, sir.
and then share your screen.
1 second, sir.
Yes. So I'll request our participants to please be patient and participate in the discussion. So what happens? All of us know. Okay. Yes, fine, sir. So what happens? All of us know that what is the impact of radiation in the DNA, good or bad, both. In good way, what it does is it eradicates cancer. In other good ways, it can able to penetrate the soft tissue and can also produce a contrast. Okay. On the because of the different densities at different layers of what do you call this what do you call is soft tissue, it can also produce a contrast. It can differentiate between different tissues very nicely. Okay. So through the various kinds of scans, as you all know, PET scan, CAT scan, and all such scans, which is nothing but applications of radiation. This is very good. So one more good aspect is it can identify and kill cancerous cell. Now, how does it kill the cancerous cell? What it does is it destroys the DNA structure. Now, what is DNA? DNA is a polymer which is made of nitrogenous bases. Okay, there is a ribose sugar which is a pentose, which is covalently linked with a nitrogenous base, either purine or pyrimidine, and then subsequently it is also connected to a high energy phosphate group. So this is the basic architecture of a nucleotide. So multiple nucleotide what do you call like garland of flower, they'll pile one after another and produce a DNA. Now DNA is having all the information necessary for all kinds of what do you call functions that is taking place in human body. Okay. Now during 1990s to 2000, we have human beings have able to decode the whole human DNA, and all the human DNA together we shall call it human genome. Now human genome is something like a pile of information where in each and every one's DNA it is written at which age your hair will start going gray, at which age you can have some specific diseases, even cancer. Okay, at which age your voice will start modulating. Okay, all informations are present in the DNA. Now this is called human genome. This is the picture sir has demonstrated. Okay, now the strands are there. No, these are different chromosomes. In human being there are 46 chromosomes. Each chromosome work each function. One such chromosome is responsible for breast cancer. Okay, now why it is responsible? Because if there are some changes in this particular chromosome, that means in DNA structure, then what would happen? Breast cancer will occur. So scientists have identified, scientists like us, biomedical scientists have identified which DNA causes which cancer. So now we'll request sir to click on the second link. Sir, please click on the second link.
Just 1 second, sir.
So this is the human genome. So human genome means what? The whole lot of DNA which is present and which is very vulnerable to destruction due to radiation. So please consider the good effect, not the bad effect. Good, what is the good effect? Good effect is radiation can destroy the cancerous DNA also. Okay. So now we'll do a simulation where we'll try to eliminate a cancerous DNA. Okay. Okay. So till now what I understood? We understood that there is a human genome. Human genome means what? All the DNA information are kept inside all cells, and this information has been decoded. Okay. Now this decoding has led us to formation of human genome. Now the first link which you can click will take you to human genome. So we will not disturb there because it's huge amount of DNA, huge amount of data. So we will not download also. Now this is the second. Now this is the second part of the thing. what I said? I said that there is a gene which is responsible for breast cancer. So we will try to eradicate cancer and we will see how. So what happens? Radiation, what it does is it not only break DNA, it can also alter DNA. Please bear with me and try to understand. What happens is radiations are breaking DNA. This is fine. So this is not the end of the story. It can also able to create mutations. Mutations means change. Change means what? Consider that in DNA there is a molecule called adenosine. Okay, so radiation, what it can do is it can convert adenosine to guanosine, one more kind of salt. Radiation has this capability. Now, this kind of changing in the DNA is known as mutation. So, radiation therapy, how it kills cancerous cells, you know, it also induces mutation. It not only break DNA, it also induces mutation. So, our job is to identify which kind of mutation is good and which kind of mutations are bad. Means what? Means I am exposing iodine 131 to a particular DNA and we are trying to see whether it can induce a mutation so that the cancer can die. So this is the whole story. Okay, it may not be classical physics, but it is related to radiation therapy, exposure, and all such things. So this is the BRCA1. BRCA1 is what? If you Google it, you will find out BRCA is the gene which is responsible for breast cancer. Okay, so I'll give you a little context in this more. Many of us has heard about one Hollywood actress, namely Angelina Jolie. So how many of you have heard of her? Please unmute, or you can say in the chat box. Many must have heard about this lady. She is a fine actress and very much talented personality. But there is a story with her. She was the patient of breast cancer. But she survived because she started her therapy very, very early. Because she did a testing of her gene and she found out in very young age that there is a possibility that she will get a breast cancer. Okay. So everyone can Google the story in internet and you can find it out what exactly happened to her. So this is the gene which is responsible for breast cancer. Okay. So in what happens if you have to eradicate breast cancer by radiotherapy? What do you have to do? You have to induce some mutation so that there is a change and eventually the cancer will die. If your radiation is not able to induce the change in this particular gene, so you can see here, gene doesn't look like this. No, this is a program. Okay. Now this program should be changed into a program which will able to eliminate this breast cancer. So our job is to find out which radiation source is good to induce this mutation. Okay. So I do not know how much you can comprehend, but we will continue to do the hands. So what we will do is, this is a complete full program, but we will change the format a little so that it becomes more understandable. So initially what I said, if you can remember, DNA is made up of 4 different types of salts. One is known as adenosine, one is known as guanosine, one is known as thymine, and another is known as cytosine. Okay. So now what we will do is we will convert this whole picture into more usable form. So I'll request our coordinator sir to scroll up. Now we are at the bottom. Now please scroll up, and then just after the name of the gene BRCA1, you can see one blue color this thing, FASTA. Can you see FASTA? Please click on this. Please click on this. So once you click on this, what will happen is the format will change. It's coming. See. So what has happened is we have changed the format of the program. So previously so much of information was there. We have reduced the noise. So this is called noise reduction. Okay. Now this is the BRCA gene. BRCA gene means what? This is the architecture of the gene. So now we will try to find out which particular radiation source can induce change in the structure so that the BRCA gene starts degrading itself and it starts killing the breast cancer cell itself. Okay, either targeted or systemic radiotherapy. Okay, so we have to find out, for example, some labs are there which will study which radioisotope can able to what you call eradicate breast cancer. Now how they will do it? They must have to change the architecture of this particular BRC. Okay, now I'll request our coordinator sir to click on the third link. So please click on the third link. Yes.
Once again, sir.
Yes, no problem. But do not close the rest.
No, sir.
The other windows are there, no? If you close, no. You have to.
Sir, can you once again send the link, sir?
In the chat box, you can. It is still. It will. Can you able to find the chat box? Shall I share the link again?
Yes, sir. Yes, sir. Sure, sir. I can get the
Okay,
links.
okay. Just. Oh, acha, acha, acha. Okay, okay. Okay. The last link, sir.
Yes.
Wait.
The first one, sir.
Why it was blocked?
Don't know, sir. This one.
Now came fine. So what has happened? Now this is the grid computer network of Harvard University Genetics Department. Okay. So here what happens? You may not have precise interest, but still I have to tell you. Softwares comes in two types. In one type of software, you have to download the software and use your computational power to do the analysis. Okay? For example, any statistical software, anything you have to download in your local computer and then do the whatever need you need. Okay. So this is one kind of software where you are using your computational power. Okay. But there are some other type of software where the calculations are so huge that you need to run it through a supercomputer. So, but if it is there, how can everybody access it? So in order to access it, what they do is they put it into a server. Now, what is a server? In server, what happens? Whatever instruction you send, no, it will get processed somewhere else in Santa Clara, Florida, and then the result will come to you. It is exactly same like your email. In email, what happens? Whatever attachment you do, no, you do not save it in your computer. Do you do? No, you do not. It gets stored somewhere in the remote server. Whenever you need it, you click it and it get downloaded in your what you call desktop, isn't it? So this program is something like this. It runs in a supercomputer and therefore you cannot able to download it. But you can use the what you call functionality. So we will do a functionality here and we will try to predict what is the impact of a mutation that we think that has caused by a radioisotope. Consider that there is a patient of breast cancer and she is undergoing one kind of what you call radiotherapy. Now, what will be the impact of radiotherapy? Can we able to predict? Now, this is a first-hand experience for all of you to understand how computer simulation helps. Okay. Now, this program runs through a mechanism called supervised learning, as I have said. So there are many classes of supervised learning. One such class is called naive Bayes algorithm. Now, this is a probabilistic classifier method which will predict a result based on previous occurrences or previous experiences, exactly like human. So this full program runs on a database which is updated every day, every moment. Okay, whenever some hospital identifies a particular cancer and identifies which kind of mutation is responsible for this, they update this particular server. This is free to use. All hospitals use this particular software to identify the impact of a mutation on any gene, not only BRCA. So we'll do an experiment with the BRCA. If you Google BRCA, you will find out it is the gene which is responsible for breast cancer. Okay. So what we'll do is we'll go to the second link. Have you closed it, sir, or it is still there? The
So
second link.
one second.
हाँ और एक बार वो second link आप on कर दीजिए. हाँ. Shall I share, sir, the link?
So the link is there, sir. It's just opening, sir.
Okay, sure. Thank you. Yeah, it will take a little time. Fine. This is the whole lot of information. We don't want that. We will only want the FASTA. So please click on the FASTA.
1 second, sir. Yes.
Yes.
It's
So
loading,
this
sir.
is the architecture. This is the architecture. And now we'll do a hypothetical. Okay, for example, as I have said, that rubidium can cause a change. So we'll see what is the impact of that change, whether it can destroy BRCA or not. Until and unless BRCA is not destroyed, your cancer is not eliminated. As simple as that, okay? You Google it, you will find it out. So what happens? We have to eliminate the BRCA in such a way that the cancerous metastasis is destroyed or gone. So what is the way? You have to use radiotherapy, chemotherapy, or some other therapy. We are studying radiation, so we will be talking about radiotherapy. So now this thing is sample is ready. So now I request you to copy the whole thing. Okay, from L you can see one L is there. No, L letter is there.
Yes, sir.
The full thing you copy.
Both lines, sir. 2 lines.
To that. Now you copy.
Yes, sir.
And then go to the server. Yes. Here
Yes,
you
sir.
click. And in the second box you paste.
Yes, sir.
Very good. Thanks a lot. Now what is the sixth amino acid? There is LTKDRM. LTKDRM. 1, 2, 3, 4, 5, 6. The sixth amino acid is M. So what we'll do is we'll search for M. Can you see AA1? AA1 is there, no, sir?
Yes, yes, sir.
Here you find M. M
Yes,
is
sir.
there. Please click on this. Now what happens? Let me tell you. So M is the sixth amino acid. So rubidium can able to change M into something else. So we will see. I'm giving you a hypothetical example. We do not know what rubidium exactly does. Okay. Consider that some hospital has said that if I give a dosage of rubidium, now this is the impact. What is the impact? M will change into something else. Now why I have taken the sixth amino acid? Most of the time, sixth amino acid alteration is enough to kill the whole jar of cancer. Okay, sixth amino acid. So it is present where? In the DNA. It's a long story, but we are cutting it into a short. So we will convert M or methionine into something else. So we will go to AA2. Can you see AA2, sir?
Yes, sir. Yes, sir.
In AA2, we'll click on A, the first amino acid.
Yes,
A
sir.
is alanine. So what happens? Consider that rubidium has the ability to change methionine into alanine. So what would happen? And we'll try to see what exactly happens and what is the impact of rubidium on BRCA1. So what is BRCA? BRCA is the gene which is responsible for breast cancer in human. From where this gene came? From the human genome map, where human genome stays inside the nucleus. Now we'll run the simulation. Submit query, sir. Submit query. Okay. और एक बार क्लिक कीजिए, देखें देखें क्या बोला. Look here.
First line.
Okay, fine. Sir, please go to the second link only. BRCA के उसमें जाइए. Now copy from the greater than sign.
Okay, sir.
Copy from the greater. Now copy and put it there.
Okay, sir.
Okay, great. Fine. Now submit query, sir.
Please
Okay.
enter submission position of protein sequence, sir.
Position in the what has happened? Please enter substitution. Okay, substitution position. Again, you go to M. It is already there, no? Okay, click on M and then click on A. Click on A
Yes.
first. Click and then submit. What they have said? Please enter substitution. Oh, अच्छा. Okay. Sorry. अच्छा, position जो है, देखिए one position. Here you put 6. The sixth amino acid, no?
Yes.
6. Submit query, sir. Okay, now here you see grid gateway interface. Can you see grid gateway interface?
1 second, sir.
Now, grid
Yes.
gateway interface means what? It is the grid computer. में सुपर कंप्यूटर में चला गया अभी. वो अभी रिजल्ट कैलकुलेट कर रहा है. देखिए लोड बहुत हाई है उसका. हेल्थ कितना है? बहुत अच्छे से 80% कम कर रहा है. कितना अभी पेंडिंग है? 1349 कैलकुलेशन है पेंडिंग. सारा दुनिया भर का लोग इसमें काम करता रहता है. फ्री है ना ये? बहुत ही प्रिसाइज है. चलिए एक बार रिफ्रेश करते हैं. So there is a refresh button, no? Please click on this. Okay. Result has come. So go to the result window. Don't go to the error. See view. Result में view बोल के है देखिए. इसमें क्लिक कीजिए. See here. See, now read out what is there. This mutation is predicted to be positive, possibly damaging. Okay, and what is the percentage? With a score of 0.504, sensitivity is 0.8, specificity is 0.9. It's excellent result. So what does it mean? It means that if a particular what do you call radioisotope can at least cause the change in terms of the sixth amino acid from methionine to alanine, this is enough to eradicate at least one single cell where this BRCA is there. Understood? So, and we have not done any high-end activity, nothing. We only did run some simulations. Now, this is how it can help us at least to predict the impact of mutation for good, good applications. Okay, and we are doing it through artificial intelligence or machine learning. Very simple concept. All the scientists in the world, they are putting data, and the computer, what it is doing is it is just calculating a possibility. Okay, so from where possibility comes? From experience. So all the experience and the result is coming to us free of cost with high precision. All hospitals, any top level cancer hospital you go, all the cancer specialists know about this particular tool. Okay, if they do not know, they'll send the sample to the diagnostic company, and diagnostic company will be able to do this. For example, Reliance Life Science. The many in Tamil Nadu, there are very good companies like LifeCell, and then what else? Then there is some many good companies are there in South India who are doing excellent in this particular area. Some universities. these days, no, these are new in all aspects. In healthcare, in every aspect, the challenges are very new, and therefore a collaborative and interdisciplinary approach is needed. This is what I think. So this is the small hands-on, and I'll go back to my what do you call presentation. There are some slides which are left, so you can preserve these links.
Shall I
Okay.
close this link, Professor?
Yes. Now I'll share my thing, and I'll go back to this my discussion. Okay. There are some these things left. Okay. So what I'll do is I'll go here, and then I'll search it. Ah. Okay. So in this hands, what we did, we understood what is genome, a human genome, and then we understood about the BRCA gene, and then we did the hands-on. So we are actively working on these what do you call applications of machine learning and artificial intelligence for biomedical research. On 2014, we started our journey, and we first published our research paper on a heart disease, which was being published and being circulated. Also, there is a genetic disease which is known as Barth syndrome. There is a NGO which have given fund for this first research that we did. Subsequently, we have developed our in-house tool and we submitted our patent, and we are very much expecting that patent for this particular pipeline mechanism will be granted, and we will also able to generate some what do you call interest among our peers. We are also speaking with some MSME of northern India so that we can monetize the product through them. There are two NGOs through which we are doing it. We have also proposed, we have also submitted a part of this whole study to a innovation challenge in IIT Delhi. The first round is clear, and we are expecting that some of our students will be called for the demonstration there itself. At the same time, a second part has been demonstrated to IIT Bombay. This is again an innovation challenge, and we are expecting to get some good results. So we demonstrated a first video where there was one giant which was moving, isn't it? And it was a film called Godzilla. So what has happened during 2013? I think 2011, 12, 13, in any of this period, there was a disaster in Fukushima. Fukushima in Japan produces radioisotopes for therapy as well as it produces fuel. Okay. Now there was a disaster in Fukushima. That particular film came out just 1 year after this, and it started in Fukushima. If you can remember Godzilla, it starts in somewhere in Japan. There is a disaster in a nuclear plant, and they find out that it resulted into a formation of a giant due to a mutation. Okay, so mutations are good and bad. Radiations are also good and bad. It is upon our students, because at the first place, all of us are teachers, and we have to let our students understand the good effects as well as the bad effects, so that they can take an informed decision. And the impact of teacher is very, very high. Only we can guide our the next generation so that they become good innovators, but innovators which are having some a mental setup to give back to the nation and to the society. So they should not indulge in something destructive work, which radiation exposure can highly lead to. If somebody is working on radioactive materials, they should only focus on the good one. So by saying so, I would conclude. Oh, there is one more important thing that I would like to demonstrate. I think many of you are already aware. So these are the major uranium deposits in India. Okay. So all of you already know. Other than uranium, also there are many radioactive materials. But let's just talk about this. Here, what happens? the radiation, what do you call, the uranium mines are located in these places. If we see this thing, what do you call, cancer map in India, okay, it is an old one but still relevant. If you look at the cancer map, no, the dark orange color is the area where there is huge impact of cancer. Okay, various kinds of cancer. So if you correlate this with this, no, there is an alarming story. What is the alarming story? That where are deposits are there, so there may be an possibility that in natural environment also some exposure is there, and therefore along with the lifestyle is also responsible, but the background radiation uh, which the people are exposed to is also important. So we should take necessary precautions, as well as stringent guidelines should be there for any kind of mining. In my area, Northeast, there is a huge mining of sandstones as well as coal. So the coal factory laborers, they are also very much prone to many cancers. So one reason is this, because in Meghalaya also, if you can remember, there is a huge uranium plant, I mean, uranium deposit, because it cannot be, the centrifuges are not deployed, and therefore it cannot be collected in a rapid way, but deposits are there. If you are having a good centrifuge, we can deploy this thing, purification plant also. So in Northeast, in Meghalaya, there is a huge burden of cancer in Meghalaya, and then some parts of Assam also. In Tripura also, it is getting every day increasing. So these are very alarming things, and we have to inform our students to take up challenges and projects which are good for the future as well as good for the society. By saying so, I would conclude, and I'll forward my sincere thanks to the coordinator as well as the institute for giving me the opportunity to discuss. If there are some questions, I will try to answer. If there are some specific questions, I will definitely try to answer.
Dear participants, the session is open for queries, interaction. Please participate.
I hope it was audible to all of you and visible.
Yes, sir, it was perfect.
Thank you. Thank you. Thanks. If any of you want me to this thing repeat, no, some parts, if you are interested, we can also able to do the repeat if there is some repeat needed. I understand that it is not classical physics, but this application part may be relevant for your research also.
Yes, participants, please interact. And this is the time for interaction before you could close.
What is the
Okay, we-
scenario in Tamil Nadu, sir, about the background radiation, or what do you call, what is the scenario of cancer in Tamil Nadu? Which cancer is most relevant in this area? Coordinator, sir. If
We are much aware of these things,
some,
sir. Yes, sir.
some participants, if they are this thing is having some idea, they can also discuss. which kind of radioisotopes are regularly being identified? If these informations are there, if anybody can share that in natural conditions, for example, nearby a river, which river is more this thing prone to having this background, what do you call, radioactivity? If there are some studies, if any of your participants are interested, they can discuss.
Participants, please interact. This is the time for interaction. If there are no queries, we shall go for the closing session.
Okay, sir.
It's a pleasure to have you, sir. Your kind presence in this program encouraged us a lot. The way you explained the clinical applications on radiation and the hands-on sessions were really useful to us, sir. Thank you for sharing your expertise, sir.
Thank you.
Thank you, participants. We'll close the session for today and we'll open up tomorrow, 7 p.m. with a third session. Thank you all. Thank you, sir.
Thank you. Thank you, sir.